Hello, everyone. My name is Ravindra Kolhe. I'm a pathologist and professor of pathology at the Medical College of Georgia. Welcome to the Bionano Symposium 2024. Today, I'm going to talk about some biomarkers which are very novel and we're able to find by utilizing optical genome mapping and investigating cancers. These are my disclosures. We have been using optical genome mapping in my lab for 2+ years. And then we have been using pretty much every sample we can get our hands on, peripheral blood, solid tumors, amniocytes, products of conception, cell lines, and we have approximately worked on about 1,000 samples, which have resulted in multiple publications. And this is just to give you a highlight of some of the publications in our lab, which goes over the plethora of work which we have been doing for the past 2+ years. We have few tests which are being used clinically, the postnatal whole genome structural variant test, the prenatal, as well as hematological malignancies. What I'm going to talk today is what and how we have used optical genome mapping in investigating hematological malignancies and solid tumors, and what kind of biomarkers we were able to identify and reveal by using OGM. So just to give you a background, for investigating hematological malignancies, we are being typically using karyotyping and FISH in a targeted panels. But the challenge with those techniques is it does not do a good justice in identifying the structural variants that well. They work very well with small nucleotide variants, but using newer technique is what we were looking for. I mean, even if with this particular combination of short-read sequencing, karyotype, and FISH, 45% of the myeloid tumors still end up being what we call as normal. The other aspect is if you look at the guidelines for coming from WHO, there's a pretty significant movement towards making hematological malignancies with very specific for what tumor type they are or what structural variant aberrations they have. And then those guidelines definitely get integrated to risk stratification. So this is such a difference between 2017 versus 2022. And our challenge is that we're still using the very old, I would say, methods to look at these updated versions of risk stratification as well as WHO guidelines. We do have now an international consortium of OGM in heme malignancy, which recently published papers on framework for how to use optical genome mapping in hematologic malignancies. What I'm going to do today is go over a couple of case study examples to show how integrating optical genome mapping is making us help do this job better. This is a case of myeloproliferative disorder where karyotype detected loss of chromosome Y, and the FISH panel was negative for any marker tested. Molecular testing detected 3 variants, but when we did optical genome mapping, in addition to detecting the loss of Y, we identified a loss of 4q deleting TET2 gene. When we integrated this data of NGS as well as OGM in the VIA software, we were clearly detecting a compound heterozygous event of a biallelic loss of TET2 with the deletion of one allele and a frameshift variant on another. So combining these two techniques and using the data coming from two different sources, short-read sequencing NGS, type assays as well as optical genome mapping and combining them together in the VIA gives us such a great visibility to keep us up to date with the new requirements of biallelic loss of TET2 or biallelic loss of P53 kind of biomarkers in hematological malignancies. Another case is in CLL where this is a case of CLL showing several aberrations, but I wanted to draw attention to chromosome 22, which is reported as di2121. To confirm a FISH was done using RUNX probe, and we can clearly see 3 signals, which confirm the 3 copies of RUNX1s, hence the chromosome 21. What we ended up doing by using this particular sample on optical genome mapping, on running this case, we not only we cannot see the chromosome gain on chromosome 21. However, we see a translocation between chromosome 1 and 21. And when we look at more closely on the structural variant view, the translocation results in breaking the RUNX1 gene, explaining the signal within the fish. So here is an example where optical genome mapping corrected the false positive call on karyotype and fish. So when we look at structural variant in plasma, this is just to give an example of IgH and what are the multiple of the partners IgH can have in multiple myeloma. If we have to follow fish panel, literally you're going to need 50, 60 different fish probes to identify each and every partner. Optical genome mapping pretty much helps you determine any or all the partners associated with IgH in multiple myeloma. So this is another example when we were able to correct the structural variant in plasma cell myeloma. In this case, the fish testing shows a gain of copy number 4P FGFR3 in 23 out of 50 cells. And when we did OGM, it revealed that the gain observed in the fish was because of the translocation 4-5 with the breakpoint at 4Q6.3. That's because of the overlap beyond these two cases. This is another case of optical genome mapping in multiple myeloma, where we are actually looking at CD138 isolated plasma cells. So this is a case where this is the same sample, same patient. The first OGM is on the sample, which is without 138 isolation, and the second one is the same sample with 138 isolation, where we can, in the original sample, we can see the OGM runs several additional structural variants, but when we do CD138 isolation, the same sample, the number of structural variants were changed. And this helps us to make sure that whatever the abnormalities we are looking at are coming from the plasma cells rather than the bone marrow itself. And this is another case which is exact opposite, where the CD138 loses, isolation cells loses any of the abnormalities which were shown on the original plasma cell myeloma. I want to move to solid tumors just to show you how we are using optical genome mapping to look at different biomarkers in solid tumors. And one of the best examples we have looked at is glioblastoma as an adult brain tumor. Again, if you go back to the new way of looking at the number of abnormalities in adult brain tumors is increasing exponentially. And this is just an example of how complex and how meticulous the algorithms for investigating adult brain tumors is. Unfortunately, we're still using very traditional methods like FISH and microarrays to look at some of these abnormalities. And I think, and then similar to heme malignancies, all these aberrations definitely help with making distinctions in subclassifying glioblastomas, which play a critical role in progression as well as therapy selection. So yeah, this is a case when we used optical genome mapping and in a adult brain tumor glioblastoma, and you can in this we compare the copy number tracks with a traditional method like OncoScan, which is microarray, and you can clearly see the optical genome mapping not only confirm what we found on a traditional method, but did way better in identifying new biomarkers. The top panel is optical genome mapping, and the bottom one is OncoScan. And here you can clearly show the concordance between 2, 6, 7, 10, 14, 19, and 22 chromosomes. And this is the same case where not only there was concordance with the standard of care methods, but we had a huge amount of additional biomarker information captured by optical genome mapping in this particular tumor. And this circus plot, I mean, if you want to look at more carefully, it shows the copy number track, the site to band, structural variants, translocation, and aneuploidies, and pretty much each and every structural variant class have additional information at a biomarker level on top of what we identified or had a concordance with the standard of care methods like FISH and OncoScan. So this is a very powerful tool which helps us identify new biomarkers in adult brain tumors. And if you want to go more into detail, and then this is the same case where we had a mosaic homologous deletion of CDK and 2A, which was also called by OncoScan, but the circus plot here shows how the chromosome 7 and 9 have an unbalanced translocation indicated by optical genome mapping. And when we look at the high resolution of this nested deletion, which can only be seen by optical genome mapping, it shows a couple of things. It shows that there's a copy number of 1.3, and then there's this unbalanced 7 to 9 translocation in an added nested deletion between these. So these kinds of in-depth analysis and information can uh, only be investigated and identified by optical genome mapping. This is the same case where we are looking at MDM4 amplification, and then you can clearly see in the circus plot, and there are 14 copies of MDM4 amplification, but there's a lot more going in that MDM4 region involving translocations of 2, 12, 16, and 17. And none of this we're able to identify because of limitations in OncoScan or microarray. We have looked at uterine cancers where you can clearly see the complexity of the chromosomes and the translocations. This is a breast cancer case where we're able to find a translocation disrupting the GRBG1 gene. The couple of other additional global biomarkers I want to talk about. One is chromothripsis, which basically is chromosomal shattering. And this is another glioblastoma case in the lab where we looked at apart from all other biomarkers, when we looked at chromosome A, there was definite chromothripsis in chromosome 8, and the zoom-in view shows the chromosome is showing multiple rearrangement within the chromosome for chromothripsis. So this kind of information and visualization is near impossible in our traditional methods of microarrays and karyotype. And in addition to that, we can go a step further by not only identifying and confirming chromothripsis, but in principle, we can reconstruct the entire region of chromotropic chromosome using optical genome and optical genome mapping. For example, by zooming into the specific region, the region around FGFR1 region, you can view the maps and reconstruct the chromosome region. So here we have reconstructed a certain region in the chromotropic chromosome that includes FGFR1 gene. From them, we can clearly map, identify two segmental duplications of P1123 and P1121 that harbor the FGFR1 gene with an inversion of an entire region. Similarly, entire chromotropic chromosome can be reconstructed to show the actual rearranged chromosome. So you can go into this much detail of structural rearrangements in a single region by using optical genome mapping. Lastly, I want to talk on HRD. HRD is a homologous recombinant deficiency. It's an emerging phenotype characterized by inability of the cells to effectively repair double-stranded break by HRD pathway or HRR pathway. It's a very emerging phenotype which is sensitive to PARP inhibitors. The important thing is we really have to look at the status of HRD to start using PARP inhibitors. Again, here's an example when we use optical genome mapping to look at HRD in a glioblastoma case. The HRD score was 32 with LSD, LOH, and TAI. And the advantage of using optical genome mapping in versus traditional methods like chromosome microarray. Here you can see this is a view in NVIDIA where we can overlay microarray data on optical genome mapping for the same case. And here the slide shows the comparisons or concordance of a call between the chromosome microarray and optical genome mapping, LOH with 2 breakdowns, which gives us a score of 3 on both the platform. This is another example of concordant call for TAI with a score of 2 in both the platforms. But the high resolution of optical genome mapping comes from, this is an example to show you. This is a 5.3 MB deletion, which is in both the chromosomal microarray as well as OGM. The 5.3 MB deletion is excluded from HRD calculation in microarrays because it does not meet the minimal threshold. But when we look at this deletion more carefully, we see two translocations that intact explain this deletion, and these two translocation definitely contributes to the HRR pathway as well as the calculation towards the score. So here we are trying to show that optical genome mapping is pretty sensitive, highly sensitive for looking at HRD. So in summary, assessing the tumor's overall structural weight and burden, definitely optical genome mapping makes a huge contribution towards it, and it's helping us to understand uh, tumor biology and hopefully help us design better treatment plan. The risk stratification correlates more accurately with optical genome mapping and in comparison to standard of care methods. I would go ahead and say that it is really helping us to identify new biomarkers, which is going to help us better classify these diseases, both in AML as well as solid tumors. In the end, I want to acknowledge my lab, who have done pretty extraordinary work in using optical genome mapping in heme and solid tumors and prenatal and postnatal samples, and Dr. Chobe, Hasty, Pang, and Sol from Bionano, which are helping us with some of these techniques. Thank you.